Thank you very much for the opportunity to tell you about our work. And today I'd like to tell you a little story which is based on a family of proteins that I'll talk about called the SREBPs that control gene expression. And these are understood in mammalian cells to control synthesis of cholesterol and fat. And our approach is very much a basic science, discovery-based approach. And I'll tell you a story about how our work in fission yeast, or in fungi, has told us something new about this family of proteins, and then how what we discovered in this model system has informed some possibilities in the clinic relating to potential antifungal drug development, as well as a little story about cancer at the end. So my laboratory is primarily interested in understanding how cells control cellular metabolism. And you can think about metabolism as all of the reactions that go on within a cell. And it's absolutely critical that these different reactions be tightly regulated in order to maintain homeostasis. And homeostasis is the ability to control a system in order to keep it stable or constant. And an everyday example of this would be the heating and cooling system in your house, where the metabolic reactions would be either the furnace that controls heat production or the air conditioning that controls cooling. And these two need to work together in concert to maintain tightly the control of the temperature in your home. And I'm particularly interested, and I won't get into the details today, but about how cells actually measure the temperature. So something in this system has to absolutely measure that temperature and say, if you want it to be 68, well, it's currently 70, and therefore we need to turn on the air conditioning and cool things down. So this control of homeostasis is important, and it's important in normal physiology. This slide shows a section of a human liver where you can see blood comes in through the hepatic artery, and this artery provides blood and glucose to the different cells. And as the blood flows from this entry point down to its exit point in the vein, these nutrients are consumed. So oxygen concentrations, for instance, are higher and experienced at a higher level here than they are by cells as it's exiting, because oxygen is consumed. And so in order for the cells along this path to do all of the same metabolic functions, make the same products that are required for cell growth, they have to handle this in different ways. This is also true in examples of pathophysiology, and this is in the development of, say, a solid tumor, where unlike normal development, where blood vessels are laid down in the proper places to supply nutrients, blood vessel formation in a tumor is very haphazard. And this means that some cells in the tumor will have high supplies of nutrients, such as oxygen, and others will be very nutrient poor. And these cells that are far removed from blood supply actually have to adapt in order to continue their growth and dividing and causing cancer. So we think by understanding more about how cells adapt to these different situations, we could find ways potentially to target and disrupt their growth. So our laboratories focus specifically on how cells regulate this homeostasis in the context of the production of cholesterol and fat. And like many of these biosynthetic pathways, there are many inputs. So there are building blocks that are required to make these different macromolecules. And they flow through a series of complicated enzymatic reactions that we may or may not remember, or we're most of us are fortunate enough to have forgotten, to lead to the production of cholesterol and fat. Now these get a very bad rap, right? High cholesterol is bad for you, fat is bad for you, but these are actually essential building blocks of all cells. But it's important that, since they're essential, you have to have enough, but too much is bad. And the cells have developed ways to control their production. And this is through this family of transcription factors, or proteins that control expression of genes in cells, that are known as sterol regulatory element binding proteins, and I'll just call them SREBP here. So when the cell senses that they need to make cholesterol and fat, they turn on SREBP, and that turns on this whole production line here to make cholesterol and fat. And when the cells have reached enough of this, they sense that, and they shut this off again. So we're interested in understanding more about how this system is controlled so that we could try to perturb it in these different disease states. And just as an example of how robust this system can be, this next slide shows the picture of a mouse liver on the left that's from a normal, healthy mouse. And on the right, a mouse liver that has high levels of these SREBPs that drive cholesterol and fat production. And this is basically mouse foie gras. So it's this completely luscious little, not that you'd want to eat a mouse liver, but it's basically that. You know, it's enlarged, it's engorged with fat and cholesterol. So this is to show you that when you have too much, that's bad. And this also plays a role in disease, where on the top is a mouse that's a diabetic model. It's a type 2 diabetes model where there's high levels of glucose in the blood. This glucose is converted into fats, which are stored in the liver, and ends up leading to a very common disease that's known as fatty liver. And on the bottom is a mouse that has this same metabolic syndrome, but has had a component of this SREBP pathway removed, and that returns the liver to normal. So you relieve the fatty liver when you knock out or remove this SREBP pathway. So these are different reasons why we're interested in studying the SREBP pathway and understanding more about the details. So I want to tell you two quick points here. First, that our studies with this yeast, SREBP, has shown that it actually responds to oxygen in fungi, and that this knowledge about oxygen has taught us something about how pathogenic fungi survive inside their mammalian hosts. And in the end, just an emerging story, or really just an idea that we have about how this information from this control system in yeast might impact our understanding of human tumor growth. Okay, so I was actually educated as my Ph.D. student in the study of basic cell biology using budding yeast, and when we were trying to understand more about this system, I thought we would take what we know about the system, which is these SREBPs that are well understood in animals, and try to understand more about their function in the context of a simpler system, and we chose this fission yeast, or Schizosaccharomyces pombe. And the SREBP pathway is broadly conserved, it turns out, in many different fungi, a few of which are shown here. And I'll be touching on a few of these today. So our early studies in this yeast basically showed us that SREBP is activated under low oxygen conditions, or hypoxia. So when oxygen drops, the SREBP pathway is turned on, and it's turned on to turn on the synthesis of these lipid pathways that are required for growth. And that's shown in an experiment here on the right, where we've taken these yeast cells and just put them in a clump and put them on an agar plate. And in the presence of oxygen, just in the laboratory, cells that are wild type grow, and those that are missing the SREBP pathway grow just fine. But when the SREBP pathway is removed and you lower oxygen, these cells can't grow because they can't turn on the necessary genes and proteins required to grow into these conditions. So the reason why this actually occurs, or how this is regulated, is shown in this pathway here that I told you before about what we know about control of SREBPs. And that is that oxygen is an important input into these pathways. So you need oxygen in order to make cholesterol. You need oxygen in order to make unsaturated fatty acids, which are a major component of fat. And so when oxygen drops, the synthesis of cholesterol and fat drops, and the cells sense this, and they turn on the SREBP pathway in an attempt to restore the production of fat and cholesterol. And that's how these yeast are actually regulating this pathway. So with that story from yeast, we said, well, how could we use this to understand more about something going on in the clinic? And we wanted to see if this was also conserved in another fungus. In this case, we studied a fungus called Cryptococcus Neoformans. And Cryptococcus is an opportunistic pathogen that infects immunocompromised hosts, and it's a major problem in areas that have high rates of HIV infection and AIDS, and it causes meningoencephalitis and pneumonia. So, and I should say, this is just a section of a human brain stained for these cryptococcal cells in the brain. So the way that humans are infected, this is an environmental fungus, you inhale it through your lungs and it gets lodged in the capillaries of your brain, and it's actually very happy there. It's a good supply of nutrients, it's warm, and it continues to grow, and you basically get these infections that lead to meningoencephalitis. So what we hypothesized is it was known that the brain had low oxygen concentrations. So we said, well, maybe it is that this organism, Cryptococcus neoformans, is going to need this SREBP pathway, which we showed in this other fungus is required to grow under low oxygen. It's going to need it to grow in this human host. So our hypothesis here was that this Cryptococcus would need the SREBP pathway in order to grow in animals or in humans. In our case, we use mice, and that this maybe would be a possible target for treatment of these fungal diseases. So shown on this slide is an experiment where we've just taken wild-type Cryptococcus and injected into the bloodstream of a mouse. And this is a coronal section of the brain showing you these massive lesions of fungus when they grow within the brain. And on the bottom is the identical experiment, but done injecting cells that are missing this SREBP factor. And the animals don't get these lesions and they survive. I could show you a viability curve, but it's, as you would predict from this, that these mice die very quickly and the mice on the bottom survive. So this actually was quite exciting because it sets up this pathway as a potential target. If we could identify molecules or drugs that would inhibit the SREBP pathway, we might be able to treat this in the context of the human disease. We were excited about this, and others were as well, and they've nicely gone on to show that in another fungus, this is Aspergillus Fumigatus, which is an emerging pathogen and an increasing problem, that this is also true. So if you, this is an example of their work. This is from Robert Kramer at Dartmouth College, sorry. That, again, when you take wild-type cells and stick them under low oxygen, they grow just fine. This is this nasty filamentous fungus. But that if you miss you're missing the SREBP pathway, they don't grow. And they've gone on to show in these animal models that the same thing is true, that if you knock out this pathway, it's also not virulent. So from that, we think it's interesting that our basic studies in this non-pathogenic fission yeast has told us something about how these two classes of widely conserved, or these major fungal pathogens might be targeted in the future. So I just want to finish by getting back to this point about how I said that tumors need to respond to this type of environment, and when they have low oxygen, they also need to be able to produce cholesterol and fat in order to divide. These are fundamental building blocks of these tumor cells. And so we thought that maybe it would be the case that tumor cells also have to respond to this need by turning on the SREBP pathway. This is not a big leap from what we were just studying in yeast, where if their environment has low oxygen, they are going to be limited for their production of cholesterol and fat, and that would turn on this pathway, which makes these products which would enable tumor growth. And so we have the simple-minded hypothesis that we potentially could inhibit this pathway as a way of slowing tumor growth. And this is, I don't have data to show you because we're just in the early stages of this, but this new technology that's known as CRISPR allows the genetic and very easy knockout of different genes in cells. And so we're working in collaboration with scientists at the Sidney Kimmel Cancer Center at Hopkins to knock out, this is a component of the SREBP pathway called SCAP, in multiple patient-derived pancreatic ductal adenocarcinoma cell lines, and seeing whether this retards the growth of these tumors in animal models. And so the idea would be, I should say that the early experiments are promising. So the idea would be that if this is the case, that under these conditions, production of cholesterol and fat is limiting, there are many FDA-approved compounds that already target these pathways. And so it may be that we could rapidly move this idea into preclinical trials. And with that, I just want to conclude saying, I tried to tell you today that our work in fission yeast on this SREBP pathway showed that it responds to oxygen and that this is important for fungal pathogenesis, and that this has led to the hypothesis that maybe tumors and solid tumors, this could have been any solid tumor. It happened that my next door neighbor, when I leaned over the fence, works at the cancer center, and he said, you should study pancreatic cancer. But it could have been any, literally could have been any tumor. And he said, I'll get you the cells next week. And that's one of the wonderful things about Hopkins is this density we were talking about. But honestly, over the picket fence, this occurred. And that I would like to take home message that, in the IBBS, it's these basic discoveries using simple model organisms that can maybe lead to transformative cures in the clinic. So with that, I'll thank some funding sources, and this is the laboratory.